Embryology is a fascinating field of study that focuses on the development of various structures and organs in the human body. One key aspect of embryology is the development of joints, which play a crucial role in our daily movements and overall functionality. In this script, we will delve into the intricate processes involved in joint development, from the initial cellular aggregation to the formation of different types of joints. Additionally, we will explore the role of genetics, molecular signaling pathways, and the impact of limb development on joint patterning. Lastly, we will touch upon joint abnormalities and disorders, highlighting their implications in clinical practice. So, let's dive into this captivating journey of joint development. In summary, joint development begins with mesenchymal condensation, where cells aggregate to form precursor structures. Signaling molecules regulate this process, leading to the establishment of distinct joint components. These events are crucial for proper joint formation. Synovial joints, like hinge and ball and socket joints, have distinct structures and functions. Hinge joints, such as the knee and elbow, move in one plane, while ball and socket joints, like the hip and shoulder, have multidirectional movement. Developmentally, hinge joints involve the differentiation of chondrocytes, joint cavity formation, and ligament/tendon development. Ball and socket joint formation requires interactions between the joint inner zone and surrounding tissues, determining joint shape. Molecular mechanisms, like WNT and BMP signaling, play a role in joint development. The joint inner zone is a key structure in joint development, formed through intricate cellular and molecular interactions. Growth factors and signaling molecules regulate cellular condensation and segregation, while cell-cell and cell matrix interactions ensure precise positioning and differentiation. Dynamic cellular movements refine joint boundaries, and extracellular matrix components provide mechanical support and signaling regulation. In the development of joints, the synovial cavity formation is crucial. It involves the differentiation of the joint inner zone into distinct joint structures, including the synovial lining, producing synovial fluid, and the articular cartilage, providing cushioning. Signals from molecules like TGF-beta and IL-1 guide this differentiation process. The synovial cavity formation requires coordination of cell differentiation, migration, and matrix remodeling. Abnormalities in this process can impair joint functionality. The fibrous joint capsule provides structural support and stability to synovial joints. It forms through the differentiation of mesenchymal cells and the secretion of extracellular matrix components. Signaling molecules regulate cell patterning, resulting in distinct layers within the capsule. The capsule also acts as a barrier against infections and maintains joint functionality. Articular cartilage in our joints is formed through the intricate process of chondrocyte differentiation and maturation. This involves the activation of various signals, including growth factors like TGF-beta and transcription factors like SOX9, which guide precursor cells to mature into chondrocytes. As chondrocytes mature, they secrete essential components like collagen type II and proteoglycans, which give cartilage its unique mechanical properties. The extracellular matrix, formed by these components, acts as a support system for the chondrocytes and helps transmit mechanical forces during joint movements. Chondrocyte maturation is regulated by the delicate balance between matrix synthesis and degradation, controlled by enzymes and growth factors. If this balance is disrupted, it can lead to impaired cartilage development and degenerative joint diseases like osteoarthritis. In summary, the synovial membrane is a crucial component of joints, producing synovial fluid that nourishes and protects the articular cartilage. Its development is regulated by various signaling molecules, and disruption can have negative effects on joint function. In summary, innervation of joints is important for sensory feedback, proprioception, and joint stability. Nerves extend into developing joints guided by molecular cues. Sensory endings detect mechanical stimuli, enabling pain sensation and protective reflexes to avoid injuries. Development relies on cellular interactions and molecular signaling for optimal innervation. In summary, the vascularization of joints is a complex process involving the formation of blood vessels from endothelial precursor cells. These blood vessels provide the necessary oxygen, nutrients, and support for joint development, functioning, and maintenance. Signaling molecules play a crucial role in guiding the formation of functional blood vessels. In summary, the development of joints in the limbs is influenced by regional specialization and signaling centers. Molecular signals and concentration gradients guide joint formation, with different types of joints forming in regions with different movement requirements. Proper coordination between limb development and joint formation is important for optimal function. Genetic and molecular mechanisms determine how joints develop. WNT and BMP signaling pathways are important in this process. WNT signaling establishes joint boundaries, while BMP signaling helps with joint formation and maturation. Imbalances in these pathways can lead to joint malformations. Joint abnormalities and disorders, such as dysplasias and congenital dislocations, are caused by disruptions in joint development. These conditions can lead to functional impairments and may require surgical interventions. Early diagnosis and understanding the underlying mechanisms are important for effective management. In summary, joint development is a complex process involving cellular interactions, molecular signaling pathways, and differentiation of joint components. Understanding these processes is crucial for diagnosing and managing joint-related conditions.